So we are constantly in a need for new cells. If you think about plants, as they grow up bigger and bigger, they're going through mitosis, replacing those plant cells. If you think about the roots of plants, if you've ever seen how they weave their way through the soil, right there at the tips of the roots is where mitosis is happening the fastest, as they're moving their way through the dirt and reaching out to get more moisture. If you shave your face, shave your legs, the act of shaving is basically damaging those skin cells that are right around your hair follicles. As you shave them off, you're basically ripping off your skin cells. And so those cells have to be replaced. Otherwise, you're going to run into trouble. You can get infections. You can have all kinds of problems. But your skin cells have to be constantly replaced. And just the act of shaving is damaging them. So you have to replace them. The process called apoptosis is where your cells all have a certain age at which they are planned to die, or we call this cell suicide. So your cells are going to commit suicide after about splitting apart 50 times, and certain cells within your body are targeted for apoptosis. And they happen in a certain progression as you go through your body, and we're going to talk about that here. But apoptosis is cell suicide, or cells pre-planning to die off. So in this process of mitosis, where we've got our cells that are being replaced by mitosis every day, we've got our red blood cells, and those are being constantly replaced. And that's very important to you being able to survive and carry the oxygen around in your red blood cells. And the rate at which this mitosis occurs can vary dramatically depending on where it is. Like inside of your bone marrow, that's where you're creating most of your red blood cells. Inside of the bone marrow is where mitosis is happening very quickly. On your skin cells, that's where mitosis is happening very, very quickly. As you're a child and you're growing and developing, making brain cells really rapidly. That's one of the reasons they say that drinking is really harmful when you're young, because if you damage those brain cells that are growing so quickly, that can have effects on the rest of your life. So children who do drugs at a very young age, or who are exposed to drugs, that's often very detrimental to them, because that's when their cells are dividing the most. So mitosis leads to these duplicate cells, and this is an example of cells duplicating. We go from one parent to two daughter cells. And so that should make sense to you. We go from one cell to two cells as they cleave and pull apart. So this is the process, and I know it's very small, it's kind of hard to see, but we have our parent cell that has all of the chromosomes inside of the nucleus. During what's called interphase, the genetic material is replicated, so we go from this set of chromosomes inside of the nucleus. We are starting to make more of those chromosomes. Then, as the linear chromosomes line up in the X's right here, they're going to begin to line up along what's called the equator of the cell. Just like we have the equator of the Earth that's right through the middle of the Earth, separating the two hemispheres, all the chromosomes line up here along the equator of the cell, and we start to have what's called spindle fibers. These fibers that are attached to the poles of the cell, and the spindle fibers are going to pull on each side of the chromosomes until at the third point right here, you can see half of the chromosomes go one way, half of the chromosomes go the other way, and they pull the duplicates apart. Then finally, since we have half the chromosomes going one direction, half the chromosomes going the other direction, the cell starts to cleave apart there in the middle until eventually we have the two nuclei that are forming around the genetic material, and we have a complete and exact copy, basically twins, of the new cells that are having the exact same genetic material inside of them. So mitosis is this 4-step process, which is what I just showed you. So here it is a little bit more in detail. In the preparation for mitosis, that's where our replicated chromosomes are there inside of the nucleus. We've got our nuclear membrane that's all around those chromosomes in the parent cell. And here is where we see these two little things here are called the centromeres. And these are the poles of the cell. If you're thinking about it like the Earth, one is going to go one direction to the North Pole, and the other is going to go the other direction to the South Pole. And the chromosomes that are here in the middle are getting ready to separate into these two exact copies. And so they are going to be pulled apart at this junction of the X, right in the middle. And they are going to go from being one single chromosome to two copies that are attached right in the middle. And they look like this X because they are attached at the middle at the centromere. And what we call these, the specialized vocabulary for it, is sister chromatids. The centromere is where they're attached. We've got our two linear pieces. What? Oh, I'm sorry. Bless you. I thought you said what? So we've got our centromere right there in the middle, and all of the genetic material has an exact copy. Think of them like identical sisters. They are, well, I guess it would be identical sisters that are attached. They are attached right there in the middle. They are identical replicated copies, and they're joined together in the middle, and they're being prepared to be separated apart, being prepared to be pulled from one side to the other side.